Hello and welcome to Laboratory 5, the in vitro chondrogenic differentiation of adipose-derived adult stem cells in alginate scaffolds. We'll start by discussing the motivation for this study. There are 40 million Americans living with joint pain today due to pathologies in articular and elastic cartilage. One of the most common causes of joint pain is arthritis, and the most common form of arthritis is osteoarthritis, which occurs because of degeneration of the articular cartilage. Articular cartilage is the smooth, load-bearing tissue lining the ends of long bones. And you can see that in the diagram. Unfortunately, because cartilage has such a low cellular composition, and because of its avascular and aneural properties, cartilage exhibits little to no intrinsic repair capabilities in response to injury or disease, and so that's why tissue engineering is looking for a solution. Now, the unique mechanical properties of cartilage is attributed to its composition. The main population of cells in the tissue is composed of chondrocytes, but these account for only 2 to 5% of the volume of cartilage. The rest is collagen, primarily type 2, and the collagen fibrils are intertwined with aggregates of glucosaminoglycans, or GAGs, which we learned about in the previous lab. The scanning electron micrograph to the right here appeared in Bone and Joint Journal in 2007, and it shows the dense fibrous extracellular matrix of native articular cartilage, and you can see a spherical chondrocyte located within a space in the tissue. And it's important to understand that this arrangement of tissue makes cartilage especially lubricious or frictionless, and it has mechanical properties so high that it can effectively distribute 10 times your body weight in a single joint. Adipose-derived stem cells, or AD MSCs, have become very important in cartilage repair research. AD MSCs are a stem cell population found in fat tissue, so they can easily be obtained from a liposuction procedure. And there are numerous studies showing that ADMSCs can differentiate into various lineages like bone, fat, muscle, and cartilage. In fact, the differentiation behavior of these cells towards a chondrocyte phenotype has been investigated extensively for cartilage repair. And from these studies, we know that growth factors can act as regulators for chondrogenesis. Specifically, we use members of the transforming growth factor beta superfamily of growth factors. We also know that chondrogenesis requires a 3D culture system. In other words, the cells need to exist in a three-dimensional configuration relative to one another, just like they do in native cartilage. And lastly, we know that a rounded cell phenotype is necessary, meaning that if the cells can exist in culture in a rounded shape, that helps to direct chondrogenesis. Now, through these studies, it's been established that calcium alginate beads are a suitable hydrogel in which to achieve chondrogenesis of encapsulated AD MSCs. Now, it's interesting that within alginate hydrogels, the cells will not attach to the alginate. They therefore stay in a rounded configuration within the matrix, and each cell feels the alginate surrounding it, which basically recapitulates a cell matrix interaction that chondrocytes would see in native tissue. And so because it's been established that this culture system works, we will utilize it in our experiment to study chondrogenesis of AD MSCs. Basically, we'll take the stem cells and suspend them in alginate solutions. And this cell suspension will then be dropped into calcium chloride solution to form tiny spheres that contain the cells. And then these tiny spheres, these beads, will be immersed in medium with a composition that is known to induce differentiation of the stem cells towards a chondrocyte phenotype because the medium will contain certain soluble factors. And we will compare the GAG expression of these cells to that of cells cultured within beads that are immersed in a standard medium without chondrogenic factors. Now, the objectives of this laboratory are to perform standard culture and maintenance of AD MSCs, to encapsulate AD MSCs within a 3D culture system, which are alginate beads, to use soluble factors to differentiate the stem cells towards a chondrogenic lineage, and to use a histological stain to characterize gag formation by AD MSCs cultured in medium with and without chondrogenic differentiation factors. Now, the materials that you will receive for this lab. You will receive a plate of confluent adipose-derived stem cells. You will also receive regular growth medium for the AD MSCs. This growth medium does not contain any soluble factors that direct the differentiation of the cells. It contains standard DMEM, like in the last lab, fetal bovine serum, recombinant epidermal growth factor, which is a protein that helps feed several mammalian cell types, L-glutamine, which is an amino acid supplement that is also important for keeping cells growing, and an antibiotic. You will also receive chondrogenic differentiation medium. It has a different composition than the AD-MSC growth medium. It's composed of a basal medium, which has vitamins, essential amino acids, and glutamine, a proprietary differentiation supplement developed by the company ScienceCell, from whom we bought the medium, and TGF-beta 3, the growth factor which we know from previous research to be beneficial for chondrogenesis. Besides that, you'll also receive alcine blue dye, trypsin, acetic acid solution, alginate solution, calcium chloride solution, conical tubes, formalin, 24 well plates, microscope slides, automatic pipette, sterile PBS, and a sterile 10 milliliter Loralox syringe with a 22 gauge needle tip. So just an overview of some major equipment you need for this lab: a laminar flow hood, inverted light microscope, an incubator, refrigerated centrifuge, and a water bath. Now for the procedure, your goal here is that you will be given a monolayer of AD MSCs and you want to reseed them into alginate beads for 14 days. So just to give you an idea of what will go on. So here's a short movie. What we have here are 4 confluent plates of AD MSCs. You will be receiving only one. But to give you an overview, what you'll be doing is removing the warm media from each of the plates and dispensing the used media into the waste bin. And then over top those cells, you are going to add PBS. This is basically a washing step. So here's a bottle of sterile PBS. And you'll dispense a certain volume over top of the cell monolayers carefully. The volume does not need to be exact. I would say aim for about 3 milliliters here, but there's no need to be exact during washes. So here we go, we're adding the PBS. And then you can rock the plates back and forth to make sure that the PBS is evenly distributed across the plate. And then you're going to remove the PBS from the plate. Dispense the PBS into a waste bin. And now that the plates have been washed, you're going to add 1 milliliter of trypsin to the plate. So the trypsin is in that conical centrifuge tube you see there. The trypsin is being added gently. And then the plates can be rocked back and forth to again evenly distribute the liquid across the plate. And then you will place these plates in the incubator for approximately 5 minutes. So just to recap what you just saw, you'll retrieve warm growth media and trypsin from the water bath. Make sure you clean each container before placing them in the tissue culture hood. You'll then remove the media from the plate and wash each plate with PBS. And you'll add then 1 milliliter of trypsin onto the plate and incubate for 5 minutes. Now this is the part that you didn't see in the video. After the plate has been in the incubator for 5 minutes, you'll take it out and you'll add 3 milliliters of warmed media over the cells. Then gently pipette the media up and down to detach the cells from the plate, just like you've done in the previous labs. You'll then transfer the cell suspension into a 15 milliliter centrifuge tubes, and you'll centrifuge it at 1500 RPM for 10 minutes at 4 degrees Celsius. When it's done, you should see a pellet at the bottom of your centrifuge tube. And just like we showed you in the previous labs, you'll need to remove the media, so aspirate the pellet, and resuspend it in 3 milliliters of growth media, and count the number of cells per unit volume using the hemocytometer. So whatever concentration you get, you'll likely have to dilute your cell suspension to a concentration of 100,000 cells per mil, and you'll need 3 milliliters total. As a reminder, this is 3 milliliters of cells in alginate solution, not cells in cell culture media. But anyway, here's the process. You'll start this by doing the calculations, C1V1 equals C2V2, just like you did last time. Now you're solving for V1 here, the volume of cell suspension you need for the dilution. C1 here is the concentration you just found with the hemocytometer. C2 is the target, 100,000 cells per mil, and V2 is 3 milliliters. Now once you calculate the quantity V1, you'll remove V1 milliliters from the cell suspension and transfer it to a 15 milliliter conical tube. You want to now isolate the cell pellet, so you'll do so by adding a little bit more media, about 1 milliliter on top of the cells, and you'll centrifuge at 1500 RPM again for 10 minutes at 4 degrees Celsius. This will produce a cell pellet, which you'll aspirate like you practiced before. So while your sample is in the centrifuge, you can save a little time by doing a little work. What you want to do is grab your two well plates, which were provided to you. One will be designated a day 0 plate, the other a day 14 plate. You can even label the lids of each well plate with a Sharpie. And what you're going to do is transfer 800 microliters of the provided calcium chloride to basically the top half of each plate. So all the wells indicated in this diagram labeled as calcium chloride. And you'll use that later. Now, when your sample is finished centrifuging, you'll retrieve it and spot your cell pellet. It should look somewhat like the one in this picture. That's the cell pellet I got when I did this lab myself. And now you'll aspirate the cell pellet and then transfer 3 milliliters of alginate solution on top of the cell pellet, gently pipetting up and down to distribute the cells homogeneously. Now you have your alginate cell suspension, but it's in a 15 milliliter conical tube. So next you'll need to transfer it into the sterile syringe and then generate alginate beads by dispensing small droplets from the syringe into the wells with calcium chloride solution. Let me give you a little bit more detail on that. So here's a movie illustrating the syringe, which has a needle attached to it. And basically, we have to stick the syringe down the centrifuge tube to suck up the cells suspension, making sure you do it slowly and carefully to avoid damaging the cells. Also, remember that the syringe will be provided to you in sterile condition, and that's why it's okay to stick the syringe into the centrifuge tube containing your cells, and you should not contaminate them. Now, you just access your well plate, and into the wells containing calcium chloride solution, you'll generate small droplets of alginate cell suspension. As you know from previous experience using alginate and calcium chloride, each of these beads will solidify in the calcium chloride, generating your three-dimensional culture system for your AD MSCs. Now I would let your beads sit in the calcium chloride maybe for about 5 minutes or so just to make sure that they're fully formed. Afterwards, you have to remove the calcium chloride from each well with the automatic pipette. Just be careful not to disturb the beads. Next, you'll wash 2 times with PBS. That means after you remove the calcium chloride, you'll add PBS to each well, then you'll remove the PBS and add more PBS. Then you'll remove that PBS and you'll replace it with 600 microliters of warm chondrogenic medium placed over the alginate beads in wells A1 through A3 and B1 through B3. That's both plates. Next, you'll add 600 microliters of growth medium, that's standard growth medium, over the alginate beads in wells A4 through A6 and B4 in B6. That's both plates. And you can place day 14 plate in the incubator and keep day 0 in the hood. Now I know this next step might seem odd, but it's the nature of a day 0 plate. What you're going to do is take your day 0 plate, and I know you just added media to the cells, but you're going to now immediately remove the media from the cells and replace it with PBS. Then you're going to remove that PBS and add 600 microliters of formalin over the cells. Let it sit for 10 minutes to fix the cells. Do 2 PBS washes, then add 600 microliters of Alcine blue stain over the beads. Wait 20 minutes while the well plate sits in the hood covered, with the lid on. Then you'll remove the Alcine blue stain and do 1 wash with acetic acid solution. Do 2 more washes in DI water. And the next step is very important. Wait your turn for a microscope. Once you're sure that you have reserved the microscope for your team, quickly prepare your samples for viewing. What you'll do is, using an automatic pipette, transfer several of the beads from the chondrogenic group to a microscope slide, and then place a glass microscope slide on top of that, and then repeat that step for the growth medium group, and you'll go over the microscope and take multiple pictures. Now, just to give you a visual, here's a little movie of the students removing the Alcine blue stain from the beads within the well plates. Again, you just want to be careful not to suck up the beads. And here is a visual of a student picking up the small beads with the automatic pipette. Now, this process may be a little bit easier if your beads are a little bit bigger. There's no problem with that. You also don't necessarily have to use the automatic pipette. If you prefer using a spatula or some other method, not your hands, of transferring the gels to the microscope slides, you can do so. This part does not have to be sterile anymore, as you'll be carrying the samples over to the microscope. Now, don't forget you still have your day 14 plate in the incubator. For homework, you'll have to change the media every Monday, Wednesday, and Friday for 14 days. For this process, you remove 400 microliters of media and add 600 microliters of fresh media on top. And so remember not to confuse the cells that have growth media and the cells that have chondrogenic media in them. At day 14, that'll be your second lab day, you'll repeat the analysis that you just did for your day 0 plate and make sure you save all your pictures so that you can do a good job on your discussion questions. And speaking of the discussion questions, here they are. In your presentation, you should cover, first of all, what was the effect of time on gag formation? Remember, you have Alcian blue staining from day 0 and day 14. What's the difference between these two time points, or are they similar? Also, how did gag formation differ between the cells that were cultured in growth medium versus chondrogenic medium? Did you see staining in both? Did one medium provide greater staining than the other, and is this what you expected? The rubric that I'll use for grading this question is shown below. You can go over that in depth at your leisure. Next, in this experiment, you cultured AD MSCs in chondrogenic differentiation medium, and you qualitatively examined the formation of gags. As you probably guessed, this is a preliminary data only. It serves as an indication as to whether or not cartilage-like ECM was synthesized by the cells. But there's other experiments that need to be conducted in order to show more conclusively that you did direct the differentiation of the cells towards a chondrocyte lineage. What are these experiments? Next, ADMSCs can be directed to form other cell types besides chondrocytes. We talked a little bit about this in the lecture. I want you to talk about what other cell types have they been used to differentiate into, and for each of these cell types, please explain which growth factors have been used, what ECM components are typically characterized for each cell type, and give examples of scaffolds that have been used for each type.